just as we did with the pig, talk about the mouth of the digestive tract as being kind of the gateway and an important part of the digestive tract. All of the functions that we talked about as far as food acquisition and food processing are essentially the same in the sheep as they are in the pig, with the one exception being that sheep don't produce any ptyalin, any amylase in their saliva, and so they don't have that starch breakdown, the chemical digestion activity in the mouth that you have in pigs. But other than that, everything else is very similar. However, the pig is a generalist. It's an omnivore animal and has adapted its mouth to process many different kinds of food. The sheep is a specialist, and so its teeth have been adapted to process just a single type of food, forages, high fiber grasses. And so we need to look at the teeth in sheep. They're going to be quite different from what we saw in the case of the pig. So I'm going to remove the cranium portion, the upper part of the jaw, the maxilla where the teeth are located on the upper part, and I want you to focus on the lower jaw. And one thing I think you'll be able to see quite easily is that in ruminant animals, the two halves of the jaw come apart very easily. They're held together by a ligament that gives this jaw joint here some flexibility. And that really is important when these animals are chewing. If you've observed sheep chewing or cattle, you know that they grind their food from in a kind of a circular or clockwise, counterclockwise kind of motion. They chew kind of laterally, and having flexibility at that joint allows that to occur. So that's the first major difference. The other thing I want you to notice, and I hope you can see this clearly, the front teeth, one of them happens to be missing. I think, can you see that there's an open slot here for a fourth tooth? I hope that's clear to everybody. So there would be four teeth on four what appear to be incisors on each side, but in reality, that fourth incisor is really the canine. Okay, and so on the bottom, what's happened is the canine has moved up and taken on the shape of an incisor, and gives us four teeth then that have that incisor-like function. And so there is no big canine tooth that you see here on the bottom jaw, no tusk-like tooth here in a sheep. And this is true for all ruminant animals. Okay, cattle, sheep, goats. They're all going to be like that. All of our deer species and so on. These are what we call spatulate type incisors in their shape. They really are specialized to harvest grass, and they have a very sharp cutting edge at the front. Okay, then if we move back, we come to cheek teeth, just as we did in the pig. And the only major difference in these is that we have just three premolars, 1, 2, 3 premolars, and then 1, 2, 3 molars. So these animals have actually lost one of the premolars that you had in a pig. They also have a very unique structure here on their chewing surface that's quite different. It allows for enamel ridges to stick up and form very sharp cutting edges that are very effective at shearing and grinding on grass. And so if we look at this, try and put it together, you can see how these two, as this goes back and forth as they chew and grind on food, you can see these sharp edges of enamel will cross on each other and give you a very effective grinding surface. Okay? Now, on the upper side, the real dramatic difference between a ruminant animal like the sheep or the cow is that they don't have any teeth in the front. No incisors, no canine at all. There's a very tough pad of gum material, gum tissue that sits over that, and so the incisor actually comes down and makes contact with the gum here, kind of crushes and traps the blades of grass there between the teeth and the gum, and then the animal will actually tear the grass loose. This is not meant to cut through the grass as much as just to hold on to it very effectively and allow the animal to pull it then away, kind of tear it from its roots. The grinding surface on the premolars and molars on the top is very similar to what we saw on the bottom, and just as we saw on the bottom, there are only 3 premolars and then 3 molars. So this particular individual, she's an older ewe, and she's lost the first premolar that would sit right here, and so she only has 1, 2 premolars. Again, younger animals would have the third one and really have their first one sitting right here, then first, second, third, and then she has 1. That's 1 molar, second molar right here, and then the third molar. The esophagus would come from the mouth, and this is all of the esophagus that we have here, about a little less than an inch or so of esophagus. It empties into this point here in this stomach complex. All of this is stomach. The stomach in ruminant animals is divided into 4 compartments. And you can actually see these quite nicely here. Beneath the omental membrane and then sticking out here is the rumen. And that's the major compartment, the largest compartment of the stomach, and it's the major site for microbial digestion. And so that would be sort of in one direction the food material could go as it comes down from the esophagus. The other way the food could go would be down in this direction into this compartment. It's called the reticulum, second major compartment of the stomach. This is also a site for microbial digestion, and it's got some unique internal structure to it that helps keep large particles of food from moving very far, any further down the tract. So recently ingested material that hasn't had a lot of time to be digested is restricted up here to the reticulorumen complex. And again, this is where microbial digestion goes on. So in a sense, you can think of this digestive tract as being just the opposite of the pig's. Microbial digestion in the pig occurred last, at the very far end of the digestive tract, whereas in a ruminant animal, it occurs right at the beginning, up front. All food material is going to be subject to microbial digestion first in these 2 compartments. Then, if the food material is small enough to pass through the reticulum, then it will move on to the next compartment of the stomach, and that's the omasum. And the omasum is really another filter, a very, very, very fine filter that, again, prevents food material from moving down any further into the tract until there is significant microbial digestion. So only the finest food particles will make it down through that omasal filter into the last compartment of the stomach, which you can see here kind of on the bottom side. And this is the abomasum. And this has the same function as the stomach in a monogastric animal like the pig or like us. This is where pepsin and hydrochloric acid are added and where chemical digestion of protein begins. This is also where that those acids and that kill most of the bacteria that have moved down the tract and actually begin to digest the bacteria as well. Okay, so that's kind of a quick look at the upper end. The rest of the structures out of the abomasum, there will be the same pyloric sphincter that separates it from the small intestine, and then you can see small intestine all down in here, liver, gallbladder, putting material, or bile in this case, into the duodenum, the first part of the small intestine, to facilitate digestion. Then, as the food material passes through all of these coils of the small intestine, it'll eventually come back. I think you can see this is the last part of the small intestine, the ileum, and it empties into the large intestine. We can see a portion of it here at the junction where the cecum and the colon meet. Okay, and we'll look at all of these parts in more detail. There's a little bit of small intestine, or large intestine. There's more of it underneath here, and then here's the final terminal portion, the rectum, and then on out. So that's just to give you some sense of what the things look like when they're all together. Now we're going to start taking this apart, and I'm going to do just like we did with the pig. I'm going to remove the omentum that surrounds the stomach, so you can see what it looks like. We'll expose the rest of the rumen. Here is the omentum from a sheep. Okay, again, you know, that same sort of lacy kind of a look that we had with the omentum in the pig. I can move some of this out. I don't know, Dave, I can kind of lay it out. Is that? So essentially the same sort of thing that we saw with the pig, the omentum, a lot of fat deposition in there, and then associated with that, must have blunt dissected it away. Here we go. And then here's the spleen. Okay, it's separated from the omentum this time when I did my blunt dissection of it away. But so there's the spleen, quite a bit different shape compared to the pig, and the pig was real long and so on. But that's the spleen. Okay, again, really a part of the immune system, more so, doesn't really have any function from a digestion standpoint. Okay, so now we've cleaned up the rumen. We can see the structure of the rumen a little bit better. A little bit of this fat off. And I think you can start to see some of the three-dimensional structure of the rumen. Again, here's where the esophagus came in. So that right there is where the esophagus comes in. And food material would first come up here. And the rumen has internally almost like a Z shape to it. Okay, there's this upper portion and what we call the dorsal blind sac, and then food material can move down here into what we call the ventral blind sac. So food material is going to stay in here for a long time and allow microbes to work on it. So what I want to do now is just open up the rumen and let you see what the inside surface of the rumen looks like, and also give you a sense of what food material looks like at this point. Remember, these animals are eating a diet that's a mixture of grain and hay and so on. And so there's what the food material looks like, and you can obviously identify some kernels of corn and other sorts of material there. But what I really want you to focus on is the inside surface of the rumen. And I'm going to try and clean some of this off. Okay, and I think that you should be able to see that it has what's kind of like a shag carpet look to it. There are finger-like projections, we call these papillae, that stick out from the surface of the rumen wall and increase the surface area there to facilitate absorption of nutrients that are broken down by the microbes. Okay? And that's very characteristic. Anywhere you go on the inside surface of the rumen, if we were to look up here, you'll see that same basic kind of shag carpet kind of look to the material. I'm going to clean that off a little. Okay, and so this is the compartment where the food will reside for the longest. It may be in here for a day and a half or so, as far as, or even a little longer as far as digestion is concerned. Okay, so that's the rumen, and again, here's the esophagus comes in, and the food material can either go in this direction or it can go to this second compartment, and that's the reticulum. And I'm going to remove the reticulum. I'm going to dump out the material that's in it. It really is essentially the same kind of material, food material that you see in the rumen. But what's really unique about the reticulum is the wall, the inside surface of it. And you see these ridges, these honeycomb, this network of honeycomb-shaped ridges. So that's what the inside surface of the reticulum looks like. And it's these ridges that trap larger pieces of food material. If you can envision this thing kind of folded back around on itself, those cells are not quite as spread out. They'll be a lot smaller, and they can catch any kinds of stems or long leaf material in that and bounce it back into the rumen so that it can be worked on further as far as digestion goes. The next compartment is the omasum, and it's very easy to identify when you're dissecting because it's a very tough piece of tissue. There's a lot of, as you'll see here in a minute, there's a lot of tissue in here that gives it a lot of internal structure. So I'm going to just cut it cross-section so you can get a look at what it looks like inside.
Looks like a chicken wrap.
We'll see. I'm going to now flush out some of the material that's in there. What you see here are parallel layers of tissue that hang from the upper surface of the omasum. Food material is going to be passing like this through the omasum, coming in, and these little sheets of tissue all have little spines on them, and they interdigitate with themselves. and create these very narrow channels that then trap very fine food material inside. You can see some of this real fine food material in here. Okay, and that then becomes a very effective filter at keeping only the smallest material, or allowing only the smallest material to move any further down the digestive tract. Okay, so very important structure, and it's what allows these ruminant animals to keep food material up there with the bacteria for a long period of time and maximize microbial digestion. Okay, and so I'm going to remove those three portions. So here we have, we've got rumen, reticulum, and omasum. Here's the other half of the omasum. And now we can look at the last compartment of the stomach. That is the abomasum. The omasum was very effective at keeping only or restricting the flow of only the finest material then out of the rumen and into the abomasum. So you can see it's quite different in its consistency. Here, much more fibrous, and here much finer particles now that get down to the into the abomasum. So there's the abomasum, and we come to the pyloric sphincter, just like we did in the pig. There's the very end of the abomasum. So as I mentioned before, this is the secretory portion of the stomach. This is where pepsin and hydrochloric acid are added to the digesta. And so breakdown of any food material that's passing down and any microbes that pass down begins here. There is the pyloric sphincter. You can see it very nicely, how much, there's kind of a constriction here, a knob here. That's a valve that restricts the flow of food material then down into the small intestine. And here's the first part of the small intestine. This again is called the duodenum, and the duodenum then is where pancreatic enzymes will be added. It's also where bile is added from the liver. Just as we saw in the pig, here's the liver, and on the bottom side of the liver is the gallbladder. We'll express a little bile from the gallbladder here in this sheep. So you can see there's some of the bile from the gallbladder. And in this particular one, the bile duct that carries the bile to the duodenum, you can actually see it right along here. It's going to enter the duodenum right down in that area. Okay. Just as we saw in the pig, the pancreas is embedded in the tissue that surrounds the duodenum. So here you see pancreatic tissue. Just lay that, just kind of lay that out there. But that very diffuse stuff, that's the pancreas, and it will also then be depositing enzymes here to facilitate digestion in the upper part of the small intestine. Okay. So, I'm going to remove the liver. We've seen everything we want to from it. Here's the abomasum and the upper part of the small intestine. So we're all done with those. Cut it off there. And we'll focus our attention on the small intestine and the large intestine. Here's the duodenum, the first part of the small intestine. Of course, the abomasum was right up here. Pancreas, liver, and bile were being, pancreatic enzymes, liver and bile being added here. And then we moved down the small intestine into all of these coils. And it is, again, just like the pig that we saw before. These coils of small intestine are linked, supported by a mesentery, and that mesentery contains the vasculature that goes up and picks up nutrients. Those blood vessels then are going to come back and carry nutrient-rich blood to the liver so that those nutrients can be processed. But you also see the fat deposition that goes on in the mesentery as well. So just like the pig, this is, we have belly fat accumulation there in the mesentery and the omentum. So other than that, things are very similar. You see the same sort of arrangement with a lot of lymph nodes here associated with the mesentery to help combat any kind of infection that might occur, any kind of pathogens that might try and invade the system through the intestinal wall. The small intestine, you can see all of these coils of small intestine here throughout. This is all small intestine here. Color really just indicates the nature of the food material that the animal was eating at that precise time. So this was probably some pasture grass or something that gives it that green color. Some of these places where it's a little bit lighter might be more of the grain mix. And then there are also places where there's very little food present at all, and so you just have this tissue kind of color. But that's all small intestine. And then eventually we pass through the small intestine, digest the nutrients that are there, and then we get down to the large intestine. And so the last part of the small intestine, again, this is called the ileum, and it empties into the large intestine complex, just like we saw in the pig. And it does it at the junction between the cecum, which is this blind-ended pouch here. So that's the cecum. And you can see the ileum then coming in from the small intestine right here. This is cecum. So food material comes in, goes either into the cecum, or it can go in this direction, which can't really see it that clearly. I'm trying to dissect things away a little bit more. But there's that junction. So, the ileum comes in, and like so. Let's lay that out. Okay, so ileum comes in right there, and it can either go down this way or it can go up like this and into the colon. Okay, the first part of the colon, what we call a proximal colon, that area. So what I'm going to do is now flip this over. Most of the colon is underneath this small intestine that you see up here. Okay, so we're going to flip that all over, try and reestablish our orientation. Here's where the ileum came in into this either cecum or the colon. And now you can follow the colon. This is all small intestine out here. This is large intestine. And this is the proximal portion of the colon. And then you can see very nicely in this sheep, and it was actually also present in the pig. It's just we couldn't see it quite so cleanly. You have this sort of spiral portion of the colon that's very characteristic of all artiodactyl animals. Those are animals that have cloven hooves, two toes. So it's true for cattle, sheep, goats, all of our deer species, all the ruminants, but it's also true for pigs. And it's a spiral portion. It's like a hairpin loop that then curls on itself. And in the case of sheep, this is the part of the colon where we get a lot of water reabsorption. So microbial digestion going on in here, in the cecum portion, the proximal colon. But then when we get into this spiral colon portion here, it's much more water reabsorption that goes on. So it passes down through these loops of the colon and then eventually comes out along here. You're actually seeing the last part of the colon, and if we can just track that down a little bit further, you get down to where I'm going to open up this portion, and we should see some very dry fecal material. Okay, this particular sheep is on a ration that doesn't appear, it's really forming pellets very nicely. You know, a lot of sheep, when it's dry, they'll have little fecal pellets, but in this case, it's a little bit more. There's a little sheep pellet. But anyway, that's kind of what the fecal material looks like now. Much different from what I think I'll go ahead and show you back in the small intestine. Or let's take it from here, where here's the ileocecal junction, or very close to it, and here's what the food material looks like there. Okay. By the time it's passed through the spiral colon and on into the lower part, the distal part of the colon, and we've had all the water reabsorption, then you can see how much drier it is, and it's actual fecal material. Okay, so I think very clearly shows you how effective that is at water reabsorption. That's it, guys.